this opportunity to give you a breakdown of what I actually did in terms of my research. So in my PhD, I studied, I used omics techniques, I used proteomics, I used metabolomics to study the plant proteins and plant metabolites. When plants were treated with bacteria, I used two types of bacteria. I used Pseudomonas bacteria species, I used Xanthomonas bacteria species. These bacteria have what is called microbe-associated molecular pattern. In my case, I use what is called lipopolysaccharide or LPS. So in my project, I use white type. White type is a plant that is intact for Arabidopsis. Then I use what is called mutants. In the mutant, I knocked out two genes. I knocked out LBRO2 gene, I knocked out BAC1 gene. So knocking out means to silence that particular genes, or it means that that gene will not be functional in those mutants, but in the white type they will be still functional or intact. This Arabidopsis plant, the white type and the mutant, were grown in the lab, I think after 6 weeks. Then we treated this plant with this LPS from this bacteria. So having treated them, we harvested the leaves. We harvest them by cutting them and throwing them inside the liquid nitrogen. So liquid nitrogen helps to quench metabolic activities. And we harvested at 12 hours, 18 hours, 24 hours. So in my first part of the study, I analyzed those plant leaves in 1D gels and 2D gels. It's called electrophoresis. And another part of the proteomics study, I used what's called LPS-induced proteomics. So in the gel-based part, it's kind of profiling the gel to see the band and spot, to be able to really see what is happening in 12 hours, 18 hours, 24 hours after this plant we assume that it was attacked by the pathogens, or can I say the bacteria. So in the LC-MS-based kind of liquid chromatography mass spectrometry, the protein extracts we are analyzed in this machine, and then using the databases we'll be able to search and then identify what really happened in the proteome level. So actually in the proteomics study, we are able to establish that this LBRO and the BAC1 proteins actually play some roles in the perception of LPS and the defense signal in plants, because we identified several proteins that play a role in defense, in transport and trafficking, and the metabolic processes, and name it. So for the metabolomic aspect, we actually use some leaf, and then we use what's called methanol extraction procedure, and then we analyze in UHPLC system. So we're able to use some databases, so we'll be able to identify as well some metabolites that play a key role in defense response to these bacteria, just glucosinolase, lignin, and lignan. Finally, in my project, we are able to really show or establish that these two proteins play a role in perception of LPS from bacteria. So I'm open for discussion, collaborations in this part of the project, because now I'm doing my postdoctoral research fellow at the same university, and I'm doing something quite different from what I did in my PhD. So thank you for watching, and stay tuned, and feel free to comment, subscribe to my channel, like, and ask questions. Thank you very much.